Hello everyone, and welcome back to AP Bio with AP Playground. Today we're reviewing Unit 7, Natural Selection. So, within Unit 7, here are the things you need to know. Natural selection, artificial selection, the different forms of genetic drift and other contributors to evolution, Hardy-Weinberg equilibrium, evidence for evolution, evidence for common ancestry, phylogenetic trees, and cladograms to represent evolutionary relationships, speciation, benefits of genetic diversity, and origins of life. So let's begin by discussing evolution and Darwin. And evolution is the change in the heritable characteristics of biological populations over successive generations. In other words, this is the change in inherited traits over time across generations within a population, descent with modification. So you're passing on your traits to the future generation, but over time, the traits that make up the population are going to be changing. And so with evolution, we also see common ancestry. So evolution happened from one common ancestor that eventually developed into all the species we see today. And we'll talk about common ancestry more later on in the video. But a population is a group of individuals of the same species that live together and interbreed. And so one of the theories for explaining evolution, and the one that is commonly used today, is Darwin's theory of natural selection. So Darwin took a trip to the Galapagos Islands, and there on each island he saw finches that had different beak sizes. And he observed these adaptations, these heritable characteristics that enhance an organism's ability to survive and reproduce. So these inherited traits that allow this organism to better survive and better pass on its genes within a specific environment. And he noticed that for each of these islands, the finches had beaks that allowed them to gain the resources that they needed to survive and reproduce for the island, depending on the food and resources available on each island. And from this, he came up with the theory of natural selection. And so natural selection explains how adaptations arise and how populations evolve. And so it begins with the idea that members of a population vary phenotypically and by extension also genotypically. So outward appearance of each member of a population is going to be different. And individuals with inherited traits that are better suited to the environment, that better allow them to survive and reproduce, they're going to be accumulated in the population over time. Because if the organism has a trait that allows it to better survive and reproduce, then that is the organism that is going to survive and reproduce within the environment. And then over time, if the environment isn't changing, then those organisms are going to make up the population, those organisms that have the traits that allow them to better survive and reproduce. And environmental change will lead to adaptation to new conditions over time. And so if the environment changes, then the trait that allows for survival and reproduction is going to become different. And so now different genetic variations are going to be quote unquote selected by nature and will make up the population over time because they are what better allow now the population to survive as a whole. And populations evolve, not individuals. So there's a change in the overall genetic makeup of a population, but that doesn't mean that the single individual changes. It just means over time there are going to be more individuals in the population that have those certain traits that allow these individuals to survive and reproduce. So over here you can see there's variation in the neck height of these giraffes, but only the giraffes with tall necks are going to be able to reach the resources that allow them to survive and reproduce. The shorter giraffes will not get these resources, and so they will die. So over time, that the giraffes with larger necks are going to be getting the resources, they're going to be surviving, they're going to be reproducing, and the population will reflect that, and over time the population will consist of these long-necked giraffes, while as the short giraffes are not going to be able to survive, not going to be able to reproduce. They're going to die, as we can see here, and so that trait will not be seen within the population. Natural selection can also change over here and take place over here, where we see these two different colors of mice, these lighter and darker colored mice, and the darker colored mice are able to camouflage. They're not going to be eaten by predators. But lighter colored mice will be eaten by predators, so they're easily spotted. And so over time, what we see happening is there's going to be more darker colored mice in the population, because those are the ones that are going to be able to survive. Those are the ones that are going to be able to reproduce. And so over time, the population will change to reflect that survival and reproduction of the individuals with that trait that allows them to better do so. And in this case, that's dark coloration. And when we talk about biological fitness, which these traits allow individuals to have, that's the reproductive success of an individual in the population, because the goal of animals is to survive and reproduce. And so if the animal can reproduce, then it has a high fitness. And while this can be tied to strength or speed, if they better allow the animal to reproduce, fitness does not equate to the gym muscular fitness that we think about within society today. It's solely based on reproductive success of an individual in the population when we're looking at biological fitness. And environmental change can affect which individual has the highest fitness. So if there's an environmental change in an individual that was adapted to an environment, the previous environment had high fitness, but now did not have the traits that allowed it to survive in this new environment, now it would have low fitness. And so when we look at natural selection, we can see that it acts on genetic and phenotypic variation. So if there is no variation within the population, then natural selection cannot occur. If there is no variation, no individual better suited to the environment than another because of no variation, then that means that the population makeup will not change, and either all individuals will die if they're not suited to the environment, or all individuals will survive if they are, but then if the environment changes, there is no chance for the population to survive. So without variation, it cannot occur, because that means there are no different traits that could be selected by nature. And more variation means there's higher chances of a population survival, because in any environmental change, there's a higher chance that there is an individual that has a trait that allows it to survive and reproduce within the population. An example here is DDT resistance, which is a resistance to pesticides. So there are going to be bugs, the insects, that have the gene for pesticide resistance, and so these, after the plant has been sprayed with pesticide, these are the insects that are going to be able to survive and reproduce. And so over time, there's going to be the majority of the insect population made up of those insects that have DDT resistance. But if there was no variation in the insect population, then there's no chance potentially there's no chance for the insect to have DDT resistance, or on the flip side, all insects have DDT resistance, but then if another pesticide showed up, then they would not be able to adapt to that, because there is, again, no genetic variation within that population. And very quickly, we'll look at artificial selection, which is the process by which species are modified by humans. This is also known as selective breeding. So instead of nature deciding which traits should be selected for, these are humans deciding which traits should be selected for. So we look at something like wolves, where humans decided to take the calmest, the most docile, the least aggressive wolves, the ones that weren't afraid of humans, and start breeding those. And as a result, we have dogs, and then they chose specific traits that they wanted to focus on, and bred individuals that had those specific traits that were prominent, and from there we get all of the different species of dog that we see today: the Collie, the Dachshund, the Bulldog, and so many others. And this is because humans decided to select for certain traits by breeding certain individuals with those traits, and that is artificial selection. Now we're going to be looking at populations as a whole. We'll be looking at population ecology, and that's going to start through population distribution. And so we have types of population distribution due to selection. There can be disruptive selection, where the extreme traits are selected for. Maybe in a population of birds, only having large and small beaks allows them to survive, and so those are going to be the traits that are selected for. There's stabilizing selection, where it's going to be that middle, that average trait that's going to be selected for. For instance, having an average baby weight is going to allow a baby to survive more than, for instance, being overweight or underweight. That's going to lower its chances of survival. But there's also directional selection, which we see sometimes through natural selection, where, for instance, there are two different colors for a trait, and one color provides more camouflage, and so the individuals with that camouflage trait are going to be able to survive and reproduce. So there's selection in the direction of that one trait, in the direction of that one camouflage color. And variation is preserved within populations. Unfavorable alleles are not always selected against. So if there's one favorable trait, then why are the other traits not removed from the genetic makeup of the population? Well, first of all, recessive alleles can be hidden in heterozygotes. So even if a recessive allele is not beneficial to survival, it can be hidden in heterozygotes that express the dominant allele anyway. And then so if two heterozygotes then breed, then there's a chance for an individual with a recessive allele to come out of that breeding. There's also balancing selection, where a mix is favored. It's actually beneficial to have multiple alleles and multiple traits within the population. We see heterozygote advantage, such as with sickle cell anemia, where sickle cell anemia is this trait that will turn your blood cells into sickles, and that will clog blood flow. And so when you're heterozygous for sickle cell anemia, some of your blood cells will be normal, some of them will be sickled, but you'll also have a higher resistance to malaria. So you'll be able to survive, but at the same time, it's going to be beneficial to you because you have higher resistance to malaria. And so the heterozygote form of sickle cell anemia is preserved within the population. There's also frequency-dependent selection, where the success of a trait is based on how common it is within the population. For instance, if you have a lot of individuals within the population that are going to have warning colors, that's going to protect the population overall, because if they're like bright colors, for instance, like poison dart frogs, then predators know to stay away from those individuals in that population as a whole, and as a result, there's going to be more survival and reproduction. So far we've mainly discussed natural selection as our primary source of evolution here, but there are other sources of evolution, and not all of them are selective. So mutations add genetic variation to a population completely randomly, and they are the only source of new genes. So they're the only way that new genes can be created rather than just selected for. And once mutations have occurred, then beneficial mutations will be selected for. Mutations don't necessarily have to be bad. They could be good if they help the individual survive and reproduce within that environment, and so they'll be selected for through natural selection. We see gene flow, another random source of evolution, which is when a population loses or gains alleles by genetic substances or additions to the population, so generally by migration, and that's going to reduce genetic differences between these populations. Over here we can see the population of yellow and the population of orange butterflies, and if a yellow butterfly comes to join the orange butterfly and they start reproducing together, then what's going to happen is now the genetic differences between these two populations have been lowered. We see sexual selection, where individuals with certain characteristics are more likely than others to obtain mates. Now this is going to be a selective form of evolution, and what this is saying is it's not enough to be anatomically compatible in order to reproduce. Individuals with certain characteristics are going to be better suited for finding mates, and this leads to something called sexual dimorphism, where there's a variation of secondary sexual traits. So maybe you have a size that is attractive, the color is attractive, there are certain attractive behaviors that help you gain mates, and so it's more than just the sex of the individual, the biological sex. So if we look over here at the peacock and peahen, the peacock that's able to strut its feathers the largest, that looks the most attractive to the peahen, is the one that is going to be able to find a mate and survive and reproduce. And so the organisms that have the best suited traits for reproduction are the ones that are going to then survive and reproduce and pass on their genes, and that is sexual selection. Another non-selective, a random source of evolution is genetic drift, which is the unpredictable fluctuation in allele frequencies from one generation to the next. And allele frequencies is how much of an allele we have within the population. And most traits that we look at, they're going to be two alleles, the dominant and recessive allele. And so when we have a change in allele frequency, when there's a shift in allele frequency, so we're changing from more dominant to more recessive, or more recessive to more dominant, that means evolution is occurring. The genetic makeup of the population is changing. And so genetic drift is the unpredictable fluctuation. It's non-selective, it's completely random, and it'll often happen in smaller populations because there's less variety to begin with, and they can lower genetic diversity because of these events. So for instance, we have a founder effect, where a few individuals become isolated from a larger population and establish a new population. So the parent population has both orange and gray butterflies, but through the founder effect, there's a chance that this new population will only have gray butterflies, and as a result, it's going to have lower genetic diversity, and there's been a shift in allele frequency. This new population only has gray individuals. We see the bottleneck effect, where a sudden change in the environment can drastically reduce the size of a population. The population has gone through the bottleneck, the skinny part of the bottle, and so where we see in this original population that has marbles of yellow, green, and red, after the bottleneck event, which is completely random, it could be a forest fire or something like that that just reduces the size of the population, we see primarily only red and green marbles left in the surviving population, and so the new population is going to reflect differently. It's going to have a different genetic makeup and different allele frequencies compared to the original population. So now we're going to go on to discussing Hardy-Weinberg equilibrium, which is how we represent a non-evolving population for a particular locus. So for a particular location on a chromosome, which is going to be for a particular gene, the population could be non-evolving, meaning that the allele frequency will remain constant. The amount of dominant and recessive alleles for that gene in the population will remain constant. And so the null hypothesis here is that the population is not evolving. And if it doesn't fit Hardy-Weinberg equilibrium and rejects null hypothesis, then that means that there is an evolving population. There is a difference between the expected allele frequency values that we can find through using Hardy-Weinberg equation, and then reality, which could be different if the population is evolving. And to achieve this Hardy-Weinberg equilibrium, to achieve a non-evolving population for a certain gene, these 5 conditions need to be met. And these are not meant to be simulating an actual environment, but this is more of a theoretical idea, the Hardy-Weinberg equilibrium, that can represent a non-evolving population. So first of all, no mutation. We cannot be introducing new genes so that they can be acted upon by natural selection. That would be evolution. There can be sexual selection. Mates have to be found randomly so that no specific individuals are selected for through sexual selection. Natural selection can also not happen because that's a surefire representation of evolution. There is change in the allele frequency within the population, and there's a shift towards a certain trait that provides a reproductive advantage. There cannot be genetic drift, so the population size has to be large so that if genetic drift occurs, if a bottleneck or founder effect happens, there is still a lot of genetic variation within the population. And lastly, no migration can happen because that will lead to gene flow. So when we talk about the Hardy-Weinberg equation, we're looking at 2 parts here. And the first we'll look at the allele frequency part. So when a gene has 2 alleles, the frequencies of those alleles are going to add up to 1. And we're going to call our dominant gene P and our recessive our dominant allele P and our recessive allele Q. And so P plus Q together, the frequency of the dominant and the frequency of the recessive allele have to equal together to 1. But the frequency of genotypes are also going to add up to 1. The entire population is going to be made up of some individuals that are homozygous dominant, some individuals that are heterozygous, and some individuals that are homozygous recessive. And that's represented by this other Hardy-Weinberg equation right here, showing that P squared for our homozygous dominant individuals, they have 2 of the dominant allele, 2PQ for our heterozygous individuals, and Q squared for our homozygous recessive individuals that have 2 of the Q alleles. Together they're going to equal to the entire population. They're going to add up to 1. And so now let's practice with some Hardy-Weinberg. And of course, this only works when the population is non-evolving, because if there's an evolving population, then allele frequency will be shifting constantly. So with a non-evolving population, you can calculate allele frequency, you can calculate phenotypic frequency using these Hardy-Weinberg equations. So you've sampled a population in which you know that the percentage of the homozygous dominant genotype, capital A, is 64%. And using that 64%, we need to calculate the following. So, first of all, we know that homozygous dominant is going to be 64%. So first, let's find our just dominant allele. Just find the percentage of the frequency of the dominant allele. So we know that our homozygous dominant, in other words, P squared is going to equal to 64%, or you can say 0.64. And so in order to get P, in order to get the frequency of the dominant allele, we can take the square root of both sides to get rid of the square and find that P is equal to 0.8. So the frequency of the dominant allele is 0.8. Now, to find the frequency of the recessive allele, it's easy. We know that the 2 frequencies together, so the P frequency, 0.8, plus the Q frequency, have to add up to 1. So we can subtract 0.8 from both sides, and the frequency of the recessive allele is 0.2. Next up, we need to find the frequency of the AA genotype. This is the frequency of the homozygous recessive allele. And so we know that homozygous recessive in our Hardy-Weinberg equation is going to be Q squared. And we know what our Q is. We know that the frequency of the recessive allele is 0.2. So we can take 0.2 and we can square that. and get 0.04. So we know that 4% of the population is going to have that recessive homozygous recessive genotype, 0.04. Lastly, we need to find the frequency of the heterozygous genotype, having one of each allele, and we know that is 2pq from our Hardy-Weinberg equation. So I'm going to do that up here. We have 2 times p, which is going to be the frequency of the dominant allele, 0.8, times q, which is frequency of our recessive allele, which is 0.2, and when we do that multiplication, we get 0.32 for our frequency of heterozygous individuals. And then we can do the math on that to check our work. If we add up all of our frequencies of genotype, if we add up 0.64 plus 0.32 plus 0.004, we will get 1, meaning we have accounted for all individuals in the population, correctly figuring out the frequencies of each genotype. So now that we've looked at different methods for evolution, we're going to discuss some evidence for it. And we're starting with direct observations. So real time we can see evolution occurring, especially with something like antibiotic resistance, where bacteria that are resistant to an antibiotic are the ones that are going to survive and reproduce, and eventually we're going to see a strain of bacteria that are entirely antibiotic resistant. But this happens very, very quickly because bacteria reproduce so quickly. So we can see it happen over a very short time, and it's a direct observation of evolution and natural selection occurring. We can also look at the fossil record, which is a collection of fossils which are remains of past organisms found throughout the world arranged in chronological order, and they show change over time by linking ancient organisms to modern species. For instance, we can see these fossils of molar teeth and these fossils of forefeet, and we can see based on their location in the fossil layer how they've changed over time, and through that scientists can build models of what different species and organisms look like over time, and we can see all the precursors to the modern horse based on fossils and how it's changed over time. Lastly, we have biogeography, which is the geographic distribution of species, and we start by looking at similarities based on location. Let's say you were in a specific area, then you're going to be sharing, even if the area had a couple of different regions in it. Let's say the area had a desert region and also a jungle region, for instance. Then the organisms within the jungle and desert region are going to be more similar than the organisms in this desert region and another desert region halfway across the world. So there's similarity based on location, even if the environment is slightly different. And this shows how there has been intermingling between these species. There's been change over time, and the allele and gene pools have shifted and mixed. We also see continental drift and breakup of Pangaea, which is the supercontinent that existed before we had our 7 continents, and once that broke up and started to move apart, that's continental drift, and that can explain the similarity of species on different continents, because at one point they could have been very, very close to each other, and even though now they're halfway across the world from each other, at one point they were very close to each other. Lastly, we have endemic species, which are species found in a certain area and nowhere else. So this shows that species have been created and that they have changed over time to adapt to their new environment, so that that species only exists in that one location. We also have molecular evidence showing that all of these organisms use the same genetic language, and we'll be looking more at that on the next slide. So some more evidence for evolution comes from homology and convergent evolution. So homology is similarity from common ancestry. So there's going to be similarity in form and structure of these organisms, even if they're used for different functions, but homologous structures are anatomical signs of evolution. That's the same body part used for different functions, but they're like this because they evolved from one certain species. So for instance, we have in humans, dogs, and birds and whales, they all have 4 limbs, and even though they're used for different purposes, for walking, for flying, for swimming, they're all going to be the same structure. These are homologous structures that are similar from common ancestry. We also see molecular homologies with lots of similar structures on a molecular level that come from common ancestry in all organisms across life. We also have vestigial organs that don't have a function now, but used to serve a function in ancestors, such as the appendix today, which has no apparent purpose, but it did before we evolved to this point where the appendix doesn't have a point, but is still existing in our body. There's also convergent evolution, where two species develop similarities over time to adapt in a new environment. So even if species aren't closely related, even if they don't have common ancestry, they will develop similarities over time because they're living in a similar environment. And these are called analogous structures, such as penguins, dolphins, and sharks, not closely related, but they all have a torpedo shape because of their environment in which they need to be going through lots of water. Same thing with bats, birds, and butterflies, all having wings because in their environments they've adapted and they need to fly. And so now when we look at evidence for common ancestry, we can talk first of all about eukaryotic cells evolving from the endosymbiotic theory, where prokaryotes were taken up by an ancestral prokaryote and those prokaryotes, such as the mitochondria and chloroplast, eventually became organelles within the eukaryotic cell. And so eukaryotic cells evolved this way, but we can also see that all of these eukaryotic cells share structures that suggest common ancestry, such as membrane-bound organelles that we discussed that came from the endosymbiotic theory, but also linear chromosomes, unlike circular chromosomes of prokaryotes, and genes that contain introns, which are non-coding regions. And all eukaryotic cells share these structures, and that shows that there is common ancestry from which all eukaryotes evolved from. And we also have evidence for continuing evolution. So not just that evolution has occurred, but that evolution is still occurring, will continue to occur. And environments are going to continue to change, and as a result, populations are going to continue to evolve through natural selection to adapt to those environments. And we see this through the fossil record that environments have changed throughout history, and we're going to keep seeing it as environments continue to change even faster due to human involvement, such as through global warming and climate change. And we see modern day evolution in genomic changes with introduced mutations, continuing genetic drift. We talked about resistance to antibiotics, and the same thing applies to pesticide resistance, such as DDT. We see direct observations of evolution happening in real time. And lastly, we have pathogens evolving and causing emergent diseases, such as SARS-CoV-2, which caused the COVID-19 pandemic. And even then, there are so many variants of it that mutated and evolved, and so we can see that evolution is continuing. It didn't just happen, it's still going on. Now we're going to discuss phylogeny, which is the evolutionary history of a species or group of species. So showing how one evolved from the other and relative age, so age in relation to one another. Maybe not a specific time at which each species evolved, but relatively to one another when this happened. And we can see this through fossil evidence, through morphology like homologous structures, genetic analysis, and molecular evidence. And looking at DNA sequences are going to be the most accurate evidence for phylogeny, because looking and comparing the nucleotides within DNA sequences are going to give us our most accurate understanding of how closely related two species are. And so phylogenetic trees are branching diagrams that represent hypotheses about evolutionary relationships. They show these relationships. And each group is known as a taxon, so sample A is a taxon, sample F is a taxon, so forth and so forth. We have branch points where there's a divergence of two evolutionary lines. So here's a branch point, here's a branch point, here's a branch point, here's a branch point, here's a branch point. And we also see the outgroup, which is the least closely related taxon. In this tree, it's going to be sample B, because there was a divergence of two evolutionary lines, and sample A, F, C, D, and E all evolved after that, but Phui B stayed on its own divergent line. And then we see sister taxa, which are the groups that are closest related. For instance, sample A and F would be sister taxa. And so they represent patterns of descent. They're not looking at phenotypic similarity, so how similar these organisms look, but they are showing the patterns of descent and evolutionary history. And the amount of change over time shown by fossils or a molecular clock, which measures evolution by rates of mutation. And that are other pieces of information that can be incorporated in order to show these patterns of descent. So practicing with phylogenetic trees, we're looking at which tree best matches the info shown in the table. So here we see nucleotide differences between species, and obviously species 1 is going to have 0 differences with itself, so these are differences, the number of nucleotides that are different in the DNA sequence. Species 1 and species 2 has a difference of 18, species 1 and species 3 has a difference of 18, but species 2 and species 3 has a difference of 9, and so forth and so forth. And we can see that species 5 is definitely going to have the most differences with all of the other species. And that'll suggest to us that species 5 is going to be our outgroup. It's our least closely related species because of how many nucleotide differences there are. And our most closely related species are going to be species 2 and species 3 because they only have 9 nucleotide differences between them, the smallest out of all of these species. So if we look for these pieces of information that species 2 and 3 are going to be our most closely related and species 5 is our least closely related, we can look through these trees and we can analyze them and figure out which one matches. So if we start on the right over here, we can see that species 3 and species 2 are certainly closely related, but it doesn't match up with species 5 being the least related, and also being the least related, but in relation to each other species, it's almost the same amount of non-relation. So it's not more, it's a little more closely related to species 1, but generally with species 2, species 3, species 4, it's all equally going to be distantly related. And this one shows species 5 closely related to species 4, which doesn't reflect the nucleotide differences. This one doesn't show species 3 and 2 as closely related or sister taxa. Neither does phylogenetic tree number 2 over here, but phylogenetic tree number 1 shows species 3 and species 2 as sister taxa, as closely related, shows species 5 as the outgroup. And if we look over here, we can see that species 1 is going to have the same distance from species 2 as it does from species 3, which we can see here because species 1 is going to be one evolutionary group away from both species 3 and species 2. So phylogenetic tree 1 is going to be the most accurate representative of these nucleotide differences. Another way of showing evolutionary history is through the use of cladograms, which depict patterns of shared characteristics, forming the basis of phylogenetic trees. So now we're focusing on similarities of appearance. Which traits do these species share? And so you place species into groups that include the ancestral species and all of its descendants, and these are known as clades. And we're looking at shared derived characteristics, so traits found in the most common ancestor and all of its descendants, but not in distantly related groups, and shared ancestral characteristics, like a trait that originated in the ancestor of the group. So all of these, the ancestor and all of its traits are going to have this shared ancestral characteristic, but a shared derived characteristic is going to appear at points within the cladogram and will include all of the descendants, but not all of the preceding species. So if we look over here at this cladogram right over here, we see between the tortoise and the horse, we got the shared derived characteristic of hair. So the tortoise isn't going to have hair, but the horse will have hair, and all of these species that are descendants within this cladogram are also going to have hair. Then between horse and wolf came the shared derived characteristic of being carnivorous, so having the teeth that can pull apart meat. So all of the following descendants are going to be carnivorous, are going to have the teeth necessary to eat meat. And so we end up here with the house cat, the most evolved out of these groups, which is going to have purring, while all of the other species in this cladogram do not. So the root doesn't have any of the traits located in the cladogram, but the most evolved species is going to have all of the shared derived characteristics shown throughout the cladogram. We're moving on now to discuss how different species are formed. And the process by which one species splits into multiple species is known as speciation. And a species is a group of populations whose members have the potential to breed and produce viable and fertile offspring. So the offspring must be able to mature and must be able to reproduce. And speciation is focused on reproductive isolation, which are biological barriers that prevent members of two species from producing hybrids, so from actually reproducing. So we have prezygotic barriers, which are going to be pre-zygote, sperm and egg fertilization. And so this is going to prevent mating or hinder fertilization, such as habitat isolation. If these species live in different areas, they're not going to be able to reproduce. They're separate species. If the organisms have different behaviors that don't allow for mating, such for instance, we have the blue-footed boobies that they have to dance with their blue feet in order to gain a mate. And if another organism can't do this, it's part of a separate species. There's no reproduction. Temporal isolation. If they reproduce at different times, different seasons, that means that no mating will happen. These are different species. Mechanical isolation. If the anatomy doesn't match up, no mating, different species. And lastly, gametic isolation. Even if a reproduction can occur, if a gamete or zygote is not formed, then there will be no offspring. There are also post-zygotic barriers, so after the zygote has been formed, but this prevents the zygote from being viable or fertile. There's reduced hybrid viability, where the development of the hybrid will cease. There's reduced hybrid fertility, where the hybrid is going to be sterile, for instance, in the case of a mule that came from a donkey and a horse. And we have hybrid breakdown, where even though the hybrids may be viable and fertile, the offspring of the hybrids are going to be weak and sterile. And this is going to show that these are different species, and speciation has occurred. So continuing to discuss speciation, we've got a few different main categories of speciation here: allopatric and sympatric. So allopatric speciation is based on geographical habitat isolation, which is going to prevent gene flow between the species and also prevent reproduction, just because there's now geographical isolation. And so we have newly isolated populations that are going to mean the gene pools are altered, especially if they're made newly isolated after a geographic occurrence. So for instance, over here we have this forest, and all of these trees are going to be part of one species, one population. But after allopatric speciation occurs, and this is generally going to occur slowly, because natural geographic occurrences take a while to happen. So for instance, weathering and erosion caused a divide within this forest. Now there's going to be more altering of gene pools, and this population has been split up, and we now have reproductive isolation and the formation of two different species. And so natural selection will occur, and as a result we're eventually developing into two different species because we split up the gene pool. So now there are slightly different variations and adaptations that are selected for in each on each side of the geographic divide, and we have isolation, so there cannot be reproduction between the two groups. Now sympatric speciation, on the other hand, is behavioral isolation. So this is isolation because behaviors don't match up, such as sexual selection, where you're not able to find a mate. If a population is switching a habitat, then the behaviors in order to gain certain resources and behaviors in surviving are going to change. Same thing if the resources change themselves, then the population is going to be differing in how they're surviving a certain environment. And over here we can see with sympatric speciation, when, for instance, there are new resources available, or this part of the population has isolated themselves from the rest of the population behaviorally, then now we have a new species, and this can generally occur a lot faster because it's not reliant on geographic events. And one example is polyploidy in plants, which is when non-disjunction, so chromosomes do not separate during anaphase in meiosis and mitosis. So these non-disjuncted plants cannot breed with diploid members that have the normal amount of chromosomes and cannot produce fertile offspring. So we have reduced hybrid fertility. But there are also hybrid zones, where these are places where species can meet. There isn't a complete reproductive barrier. And what can happen at these hybrid zones is one of three things. Either the barrier will be reinforced, ensuring that yes, these are two separate species. The species can fuse back into one, or the hybrids will stabilize. So yes, these are two species, but also hybrids can be produced and they have the chance to be viable or fertile. And evolution and speciation can occur at different rates. And so we're looking at divergent evolution, where adaptation to new habits results in phenotypic diversification. So as there's a new habitat and as populations are adjusting to it and adapting to it, there are going to be certain traits that are selected for, and eventually, because in this new habitat they're looking for different resources, there are different community interactions, there could be speciation because behaviors are going to change in result to this new habitat, and we see sympatric speciation. Now with adaptive radiation, which is one rate of evolution and speciation, many new species arise from a single common ancestor due to many new niches, so many new roles within the environment of organisms to use certain abiotic and biotic factors to be filled. So for instance, after an extinction, it's almost like a clean slate. If there's a single common ancestor, it can then generate a lot of new species because there's so many niches that are able to be filled within the environment, and that as different populations adapt to certain aspects of the environment, they're going to change their behaviors. We also see punctuated equilibrium, where there are periods of stasis, not much evolution, followed by sudden change, as compared to gradualism, where you have gradual evolution over time. And divergent evolution is going to compare to convergent evolution, which we discussed earlier, where there is no common ancestor, and there's evolution of similar traits due to similar environments. But divergent evolution is we're going from a common ancestor into multiple species. Speciation is occurring because habitats are changing, and so there are behavioral changes or geographic isolation, so allopatric or sympatric speciation. And genetic variation is super important to natural selection and evolution. Different genes are selected for in different environments, and genetic variation is what makes natural selection possible in the first place. If individuals are all the same, then one cannot be better adapted to the environment than the other. And genetic variation means that in different environments, certain individuals may have a better adapted trait for that environment. So those are the individuals that are going to survive and reproduce. And populations with little genetic diversity are at a risk of decline or extinction because they don't have this variation. So if the environment suddenly changes, they might not have an individual with traits that can adapt to it. And so this is the issue with having things like crops within the form of monoculture, where a single crop, possibly even a genetically identical crop, could be planted on a field. And so if one crop is going to be affected by disease, then so are all the other crops, because none of the crops are going to have a trait that could help them be resistant to this disease, because there is very little genetic diversity. And we get things like the Irish potato blight. We also have genetically diverse populations and ecosystems as a whole, which become more resilient to environmental change because they're more likely to be individuals that have traits which allow them to withstand and adapt to the new environment. There's a higher chance that when the new environment shows up, there will be an individual that has a trait that allows them to better survive and reproduce in that environment. And which traits are beneficial are going to be dependent on that environment. So genetic variation is super duper key to natural selection, and without genetic diversity, extinction is very possible. And we're going to end this review talking about the origins of life, so how life arose on Earth. So Earth formed approximately 4.6 billion years ago, and life first appeared 3.5 billion years ago. So prokaryotes are going to be our first life forms, our simple single-celled organisms. And once photosynthetic organisms were working enough and there was enough oxygen in the atmosphere, then cellular respiration became a thing, and well, it evolved to become a thing, and eukaryotes now began happening as well. We've got more complex multicellular organisms, and we see this evidence using geological data, dating of the fossil record. And so there are some models that predict how life could have arose. So the Opernd-Haldane hypothesis states that inorganic compounds, so compounds not containing carbon, energized by lightning or UV radiation, developed organic compounds. So organic compounds are macromolecules, and life itself came from inorganic compounds. And the Urey-Miller experiment showed that yes, macromolecules can be formed from inorganic compounds. Now the criteria for having this atmosphere was slightly different from the Opernd-Haldane hypothesis, but the important thing is that macromolecules can be formed from inorganic compounds, and depending on which inorganic compounds, what essential elements they have in them, that could determine which macromolecules are created. So for instance, we look at our Chomp rule that shows the different essential elements within macromolecules, with lipids and carbohydrates having carbon, hydrogen, and oxygen, proteins having carbon, hydrogen, oxygen, and nitrogen, and nucleic acids also having phosphorus. So by having these essential elements in the inorganic compounds, then we're able to develop these biological macromolecules, these organic compounds. But on the other hand, meteoroids may have transported organic compounds, and that's how they showed up on Earth. And so when we went from inorganic to molecules to organic monomers and then organic polymers, we eventually reached protocells, which were just membranes, essentially. They had a membrane differing the internal and external environment, and so they were not necessarily cells with any specific components, but membranes were formed, and then after that arose self-replication. And the RNA world hypothesis states that RNA was the first genetic material. RNA is what then eventually led to the beginning of life and DNA becoming the primary genetic material. And so RNA replication is what allowed for the first cell division to happen. And ribozymes, which are RNA catalysts, RNA enzymes rather than protein enzymes, are what made everything happen. So RNA was the starting genetic molecule from which life arose, and then DNA eventually over time evolved to become the most important genetic molecule. So that was all of Unit 7 reviewed, and we'll see you in Unit 8.